Okay, hello everybody. Like Janelle said, my name is Jalen Sims. I'm a graphic design major and I'm part of the McNair Scholars Program. So for my research project, I will be sharing with all of you a study on urban architectural design and how it can be better integrated with nature and sustainability. The biggest thing about this project was I wanted it to be consumer friendly and easy to understand because the people that are going to be hopefully using these are people just like you, um, who are not like major construction people and you're just average citizens. So starting at the top of this slide, I wanted to focus on ways that we can reach symbiosis with our environment. So the first thing is water. Some of the ways that we can better integrate ourselves with this abundant resource is by using pond or a water tower as irrigation for the plants in our area. Another thing we should be doing is investing in hydroelectricity because this is a great way to get energy that we need. And finally, we should think about using vertical hydroponic systems as a way to grow food because they use less water and soil and they're more productive than traditional farming. Moving on to plant life in our area, we should be planting native grasses and soils on our roofs to attract birds and also insulate our homes better. We should also have vines and ivy hanging from our roof to help offset our carbon input, and that can also be a great natural decoration for our space. Next up is wind energy. We should be investing in wind farms, and when it comes to buildings, we need to make sure that all windows are able to be opened so we can better regulate temperature without having to turn the air conditioning on. And finally, there's solar power. We should be investing in solar panels on our homes and around our cities as the way to get energy. We should also focus on building designs that have lots of windows in them to allow natural light to come in, which boosts people's mood and saves on electricity. Looking at the bottom left portion of the slide, I have found 6 different materials that are an alternative to concrete and other standard building materials. So the first one is straw bales. These are great for insulating a home and they're a rapidly renewable resource. Hempcrete is very easy to make. It's stronger than concrete. It's carbon neutral and it's actually great for farmers because the hemp, the root structure, it actually breaks up the dirt in the soil, allowing the next crop rotation to easily get nutrients. Recycled plastic or eco bricks are great because it finds a new use for all the plastic we're creating and can be very cost effective, and especially in areas that have less money and less resources. And before my time ends, I wanted to take a moment and talk about the list over on the right. I encourage you all to take a picture of this list as it can be a way for you to keep track of, or by doing the things on this list, you can increase the sustainability of your home and your life. So a green roof is when you have soils and grasses on your rooftop to insulate your home. When working on construction projects, we should keep in mind these alternative materials that I've showed you. And yeah, please take a picture. And that is my 3-minute thesis.
So our next presenter is also a McNair Scholar. This is Stephanie Baklarz. She is in chemistry, medicinal plant chemistry specifically. Her advisor is Dr. Alexander Wilson, and she'll be presenting the effect of foliar applications of methyl jasmonate on the terpene content of cannabis sativa L as quantified by GCMS.
So the effect of foliar applications of methyl jasmonate on the terpene content of cannabis sativa L as quantified by GCMS is really just a fancy way of describing the question, what is going to happen to my cannabis plants if I spray their leaves with hormones? And in this case, the hormone was methyl jasmonate. But really the big question here is, why do we actually care about increasing the terpene content in our cannabis? And if you're not sure of what terpenes are, they're just small volatile molecules that are responsible for the taste and smell of things like lavender or basil or hops, if you're a beer drinker. In terms of industry, they're all over the place. They're in the pharmaceutical industry, the health and wellness, the cosmetic, the food and beverage, et cetera. So those are all billion-dollar industries, so terpenes are pretty relevant. As far as cannabis is concerned, though, terpenes actually help increase the medicinal value of cannabis because of the way that they interact with other molecules like THC or CBD, and that's called the entourage effect. And that's basically the idea that all of these chemical constituents provide a better medicine together than they do as separate individual extracts. So the goal here really is to increase the medicinal value of this cannabis without an accompanying increase in the cost to do so. Methyl jasmonate's not terribly expensive, and it's very active physiologically at really low concentrations, so you don't use that much anyway. So to do this project, I had 4 groups, each with 4 plants, a control group which got no hormones, a group which got 1 millimolar methyl jasmonate, a group which got 1.5 millimolar methyl jasmonate, and then finally a group which got 2 millimolar methyl jasmonate. So the hormone was applied using basically a fancy spray bottle on the tops and bottoms of the leaves. And methyl jasmonate is actually known to regulate pathways which produce terpenes. So the hypothesis for this project was that the plants in the 2 millimolar methyl jasmonate group are going to have an increased terpene content relative to the control in other groups. Unfortunately, my project rather is still in progress. So I don't have direct results, but you can see here on the bottom left of the slide, there's a photo of a plant harvested out of the control group next to a 2 millimolar methyl jasmonate plant. And I'll let you try to make your own conclusions there on if you see a difference. I don't want to bias my research. Currently, what I have left to do, I need to take random samples from each group, take extracts of those samples, and then inject those extracts onto the gas chromatograph mass spectrometer, or GCMS, to analyze the terpene content. And with that, I thank you for listening.
And next up, we have Ashley Martinez. She is a McNair Scholar in the History Department. Her advisor is Dr. Robert Goodrich. And today she'll be presenting the culture of remembrance in Austria about the Holocaust.
Hi, so my research is on the culture of remembrance in Austria about the Holocaust. So the culture of remembrance refers to the policy of confronting Nazi-era crimes through the frank discussion and by acknowledging the responsibility for the Holocaust. So the Anschluss, or the annexation of Austria, happened on March 12, 1938. Then towards the end of World War II, a victim theory was adopted by Austria. The victim theory began with the Moscow Declaration that stated that Austria was the first victim of Hitlerite aggression. Then, in 1945, the occupation doctrine stated that because Austria's statehood had been dormant during this occupation, they could not be held liable for any of the crimes committed, and therefore they did not have to pay reparations or restitutions to the victims of the Third Reich, since they were also considered victims. However, this self-sacrificing myth is contradictory to this theory. A major example of this is that while Adolf Hitler entered Vienna in a militarized parade, there were 200,000 Austrians present, welcoming and cheering him on, as you can see in this image. It wasn't until the 1980s that Austria began to fully accept the role that they played in the Holocaust. The Chancellor and President in 1991 gave notable speeches admitting that Austria not only were victims, but also perpetrators. The government began some restitution initiatives. They began putting World War II memorials along with memorials for the victims of the Holocaust. They began rebuilding synagogues, reopening Jewish museums, and memorializing a concentration camp in Austria that was only 100 miles away from Vienna. So what caused this shift in Austrian government and Austrian policy to adopt a culture of remembrance? Well, ultimately, it was international pressure that stemmed from political scandals that tied Austrian politicians to the Third Reich. And through my research, I examined Austria's culture of remembrance and how it influenced governmental policies, and I highlight the positive and negative changes within the social structure.
So on the graduate side, first up is Tru Hubbard. Tru is a graduate student in the biology department, and their advisor is Diana Lafferty, Dr. Diana Lafferty. And they will be presenting Carnivore Guild Structure, the Bobcat's Role.
Hi everyone, my name's Tru Hubbard. I'm a second-year master's candidate in the Department of Biology, and I'm excited to be here today to talk about some of my research with you. I've had some amazing opportunities over the past 2 years. One of them has been to be a part of the first ever nationwide camera trapping survey to document wildlife in all 50 states, a project called Snapshot USA. If you've never heard of it, I encourage you to check it out. It included hundreds of researchers across the entire US working together to create this incredible data set that can answer an abundance of ecological questions. As a collaborator on the project, I was super excited to be able to work with this data and answer a question of particular interest to me. My question is, what role does the adorable yet very skilled hunter, the bobcat, have in this diverse US carnivore community? If you don't know, the US has 39 native carnivore species, ranging from larger apex predators such as the cougar and gray wolf to smaller subordinate carnivores such as the red and gray fox, which can be highly influenced by things that occur higher up in the food chain. Bobcats are very adaptable, medium-sized carnivores, also known as mesocarnivores, yet in some environments they have been known to act as the apex or top predator. As such, with access to this incredible nationwide snapshot data, I wanted to examine how bobcat dominant or subordinate status, as well as their daily activity patterns, shift relative to the variation in the carnivore community structure across the US. Using camera traps, a non-invasive method of capturing wildlife images, over 400 bobcat detections were captured at approximately half of the camera arrays located throughout the country. I learned that the highest relative abundance of bobcats occurs in the Southwest region, yet many detections were captured on the East Coast, where the current bobcat population range says they actually don't exist. I found this to be really interesting. Further, of the 9 species for which I had sufficient data for analysis, bobcats were found to have the highest activity level of the 9 different carnivores I compared. This means the bobcat was active for the greatest proportion of a single 24-hour day. Based on the activity curve graph that's located in the top right corner of my slide, we can see that the bobcat, the dark blue line, had a greater density during the daytime hours, and this pattern was consistent across all regions of the US. Lastly, I found that bobcats avoided both coyotes and fox, yet coyotes were actually attracted to bobcats. This may mean bobcats are trying to limit their competition as much as possible, yet coyotes may be trying to take advantage of the bobcat's resources. Ecologically, understanding the carnivore community structure and how it varies across the diverse multi-use lands of the United States is critical to the conservation and management of hundreds of species. The impact of one single carnivore species can cause cascading effects that reverberate throughout the entire food web. As such, we must continue to invest in research that can inform our development and effective management strategies in a world that is ever changing by our human influence. Thanks so much for your time.
So our next graduate presenter is Casey Juntila. She is a graduate student in clinical molecular genetics, and her advisor is Dr. Matthew Jennings. And today Casey is presenting Are You Infectious?
Are you infectious? The COVID-19 disease caused by the SARS-CoV-2 virus has affected everyone across the globe and has resulted in over 106 million cases worldwide. A person infected with this virus can display a wide range of symptoms or can be asymptomatic and display no symptoms at all. A PCR test designed by the CDC and emergency released by the FDA has been designated as the standard test used in COVID-19 diagnosis and has aided in performing roughly 312 million tests in the US alone. The sample used for this PCR test is the nasopharyngeal swab, or the little swab that goes up your nose and pokes your brain and generates results within 60 minutes. So what I propose to you today is a fast and simple test for COVID-19 disease screening. The asymptomatic population of individuals that this is targeted for is likely spreading the virus unknowingly and should instead be in quarantine. This test relies on the detection and amplification of 3 specific viral genetic targets, ORF1ab, E, and N, using a technology that is more sensitive than PCR. Using these 3 targets instead of just 1 increases the test sensitivity and specificity and generates results within 30 minutes. Asymptomatic individuals begin the testing process by collecting saliva into a sterile tube. This saliva is then tested using our new gene amplification technique, and results are indicated using a simple color change. So as amplification occurs in the presence of any viral genetic material, byproducts are released, acidifying the solution and causing a color change of the pH indicating dye. So a pink color indicates a negative test and a yellow color indicates a positive test. Using this fast, simple, and less invasive test for COVID-19 disease, screening can be more effective and help people take the proper quarantine measures, further reducing the spread of the virus. In a trial study of 47 NMU campus volunteers, including students, faculty, and staff, were tested using this new technology, and 6 were found to have asymptomatic COVID-19 disease. And to my surprise, my personal sample, which was being used as a negative control, became positive. Additional tests were performed only to confirm I indeed was an asymptomatic individual who the previous day was outside jogging throughout my neighborhood. Lab testing at the NMU Health Center generated negative results, and due to the lack in testing resources, our testing was shut down. The results from our 47 individual trial yielded a positive test rate of 12% as compared to the NMU reported rate of 0.6%. Thank you.
So the next graduate student to present today is Emily Griffith. She's a graduate student in biology department. Her advisor is Dr. Alec Lindsay, and Emily will be presenting sexual dimorphism of long-eared owls, plumage in the ultraviolet spectrum.
As she said, my name is Emily Griffith. I am a second-year biology master's student here in the Department of Biology, and I study birds. More particularly, I study bird plumage and the differences between males and females. When most people think about this, they think about birds like cardinals that have really, really bright colored red males and more drab kind of brown females. But some birds and some species like owls, you can't really tell the males from the females just by looking at them. But that doesn't really matter to me because I'm not looking at birds in the visible spectrum. I'm looking at them in a spectrum of light that we can't perceive. So owls, especially the long-eared owl, which is the species that I study, can see in a wavelength of light beyond what humans can see. Humans can only see colors or these different wavelengths in what is called the visible spectrum, but owls can see in what is called the ultraviolet spectrum, which is just kind of out of our reach. Even more interestingly is that if you look at owl feathers under a black light, they will fluoresce a bright pink, which not only makes owls the best birds to party with, but also shows that they have a special type of pigment in their feathers that we necessarily can't see. So what my research does is it looks at these feathers to see if we can individually look at these pigment concentrations to see if there is a difference between the males and the females. So to do this, I've partnered with long-eared owl banders at Whitefish Point Bird Observatory in Paradise, Michigan, which isn't too far from here. And I have taken some feather tips from the feathers they collect while banding to genetically sex the birds. And then I've used the rest of the feather to go ahead and put it into what is called an HPLC, or high performance liquid chromatography machine. This allows me to isolate the particular pigments that cause this fluorescence and measure their quantity by visualizing them through a graph like you see in the bottom there. The higher the peak in the graph, the more fluorescence and the more of these pigments that we can see. And so far, we're finding that males actually do have a higher amount of fluorescence in these feathers than females do, which is super duper interesting. And I am super excited to see what the implications of this research may be on sexual signaling and how we understand plumage coloration moving forward in the future. Thank you.
So our next student is Katie Elwell. She's a graduate student in psychological science department. And her advisor is Dr. Joshua Carlson. She will be presenting functional changes in the anxious brain cerebellum following attention bias modification therapy.
The National Institution of Mental Health reported that approximately 68% of US adults experienced high and long sustained levels of anxiety in 2020. This is crazy, but unfortunately not surprising, just considering how awful 2020 was to all of us. Now more than ever, we need an easily accessible therapeutic option for anxiety patients. My thesis, which is a part of an authorized clinical trial funded by the National Institution of Mental Health, is doing just this. I'm investigating 2 critical components, with one being the development of a new anxiety therapy called attention bias modification, or ABM for short. So this therapy is actually done right on your phone. ABM works by utilizing pairs of threatening and non-threatening stimuli to retrain the brain's dysregulated, hyperactive, and biased attention control to potential threats. So this is a hallmark symptom of anxiety. So because you can customize the pictures and the stimuli in this therapy, it allows one to focus on retraining anxiety specific to the patient. So other therapies are not specific to the patient, they're expensive, they're hard to access, and they have outrageous side effects. However, ABM is affordable, accessible, and can be done right on your phone. Other clinical trials have actually indicated that it is quite effective in decreasing sustained levels of anxiety. I'm also concerned with assessing brain connectivity patterns linked to the cerebellum in anxiety patients. So typically the cerebellum has only been implied in motor movements like walking, posture, balance. So honestly, this is crazy to me because our brain is vastly connected. The cerebellum is a massive structure and I surely thought it was capable of more. So I thought something else had to be going on here. Well, there is. So you see the participants in my study receive an fMRI immediately prior to their therapy, immediately following their therapy, which lasts 6 weeks, and then another one 1 month after their therapy ends. So these fMRIs are mapping live moving brain activity via blood flow changes. So this allows us to assess live brain connectivity before and after therapy in these patients. So essentially this is showing us what parts of the brain are dysfunctional and responsible for anxiety. It allows us to map out how one's brain is working. It shows if changes in the brain are long lasting after this therapy. So preliminary data is indicating that the effects of this therapy are long lasting. So lo and behold, there is quite a bit of connections between the cerebellum and threat and anxiety regions in the brain, indicating to us that the cerebellum does in fact play a role in modulating anxiety. Preliminary data indicates that these connections between the cerebellum and anxiety brain regions are decreasing following the conclusion of this therapy, and that this therapy is long sustained. So these findings will have huge implications for both the understanding and treatment of all neuropsychological disorders. I'm excited to contribute to the up and coming literature concerning this new anxiety therapy, as well as contribute to the lack of literature regarding the role of the cerebellum in neuropsychological disorders. Thank you.
So our next graduate student is Emily Klinkman. She is in the exercise science program and her advisor is Dr. Sarah Breen. She'll be presenting coordination, variability, and injury risk in experienced collegiate dancers. Sorry, my mouth is starting to run out here.
I'd like to begin by presenting to you a hypothetical situation. Let's say you're an athlete preparing for a performance. The day before the event, you take a bad landing and you break your foot. You're absolutely devastated and in for a long recovery. Is there anything you could have done to prevent this? Could you have even predicted it? I'd like to explore this situation by focusing on one sport in particular, dance. Dancers are athletic powerhouses who are often under immense pressure to prepare and perform. Injury is all too common in dancers, largely due to the amount of time spent in dance practice, the repetitive jumps and landings, and the hard flooring upon which they spend most of their time. In sports outside of dance, injury has also been linked to a concept called coordination variability. If we break this term down, we know that coordination is how certain joints of the body or limbs synchronize with one another to produce movement. Variability is how much the synchronization of these body parts changes throughout trials of the same task, or the standard deviation. In other sports, literature has demonstrated that coordination variability is higher in a healthy system. It also tends to be higher in more experienced athletes, and lower coordination variability tends to be associated with injury. In dancers, however, higher dance skill is associated with lower coordination variability, possibly due to the pressure to maintain visually consistent movements. There's limited existing literature connecting injury and dance and coordination variability. So the question becomes, how can we measure coordination variability's relationship with dance injury? So in my study, dancers will come into the lab and do some jump trials in a 3D motion capture field. To calculate coordination variability, we'll take positional data and plot the joints of the lower limbs in relation to one another. In relation to one another in angle-angle diagrams, and the standard deviation is the coordination variability. We will then follow dancers for 4 to 6 months to see how often they are injured. We will set up an equation to link coordination variability, injury, and other factors like how long the dancer has been practicing, how much time per week they are practicing, and how many previous injuries they have had. The goal is to determine the strength of the relationship between coordination variability and injury in dance. We expect to see a strong relationship between low coordination variability and injury, much like what we have seen in other sports. The end goal is to develop a method to improve pre-dance participation screenings. If a dancer has a high risk for sustaining an injury due to their coordination variability, dance instructors could modify training to appropriately minimize injury risk. If successful, findings could also be applied to other sports and be developed into a sport-specific screening protocol. Thank you very much.
So our next presenter is Xochitl Delgado. She's a graduate student in the Psychological Science Department, and her advisor is Dr. Amber Lacrosse. She'll be presenting fluoxetine as a novel treatment to attenuate cisplatin-induced cognitive impairments.
Hi, everybody. Okay, do you know somebody that has had cancer? Do you know somebody that has gone through chemotherapy? You probably do. In January 2019, there were more than 16.6 million Americans alive with a history of cancer. Around 30% of these survivors experienced cancer-related cognitive impairment. So you might be wondering, what is cancer-related cognitive impairment? Some people that have gone through chemotherapy present alterations in certain cognitive domains such as memory, attention, processing speed, and executive function. These are a set of mental skills that include working memory, flexible thinking, and self-control. Alterations in this cognitive domain impact everyday life of cancer survivors, as they report experiencing difficulties in undertaking and accomplishing even simple tasks like keeping track of and paying bills, or even getting ready to go out. As a result, novel therapies are being assessed on their ability to prevent or reduce the effects of these cognitive alterations. My project is interested in fluoxetine, also known as Prozac, a common antidepressant. We want to see if fluoxetine has the ability to attenuate cisplatin-induced cognitive impairment. Cisplatin is a common chemotherapeutic treatment that has been proven to produce cognitive alterations. So why fluoxetine? It has been shown that fluoxetine can produce neurogenesis, the growth of new neurons in the hippocampus, a key area of the brain. It has also been discovered that fluoxetine has anti-tumoral effects. So what am I doing? I started with an in vitro study. This is a fancy word for studies done on cells. I'm treating different cancer cell lines with fluoxetine and cisplatin. I want to determine what's the lowest dose of cisplatin that I can use to kill the most amount of cancer cells. And since fluoxetine has anti-tumoral effects, I want to accompany the cisplatin treatment with this antidepressant so we can not only kill cancer cells, but also have less side effects due to chemotherapy. I'm also doing an in vivo study. This is another fancy word for studies done in animals. I'm treating mice with cisplatin, expecting to see impairment in the mice's ability to perform a behavior task, but also treating them with fluoxetine to see if this drug will attenuate the cognitive impairment. This research has the potential to help better understand the underlying biological mechanisms involved in cognitive decline due to chemotherapy treatment, and will provide us new information on how to improve treatment and quality of life for cancer patients. Thank you.
Our last graduate student is Olivia Kingery. She is in the Master of Fine Arts and Creative Writing program. Her advisor is Dr. Monica McFawn, and she'll be presenting The Sound a Body Makes and Other Ruminations on Navigating Roadkill.
Hi. My name is Olivia Kingery, and my thesis is a collection of nonfiction poetry and watercolor images, all centered on roadkill. My thesis is a story, so I thought, why not tell a story? But it's not just my story. It's Marquette's story, it's every more than human being's story, and it's our story. I've lived in Marquette for 8 years now. I've watched our shoreline ebb and flow, but the changes that sit hardest are the ones that we don't always see, or the ones that we actively turn away from. This summer, I moved the body of a fawn from the side of Wright Street, just a block from my house. The sky was blue, the sun was shining right there on her, and within the same week, little did I know, I'd be crouched over the body of another neighborhood fawn who had met the same fate. And in my periphery, I can often see people watch what the weirdo on the side of the road is doing. They watch me move deer, squirrels, chipmunks, turtles. And recently, a couple honked and cheered as I was moving the body of a deer from where US 41 and Washington meet. As I was walking back to my car, all tears and deep breaths, I thought, I hope they would slow down for the next beam they saw across the road. And I hope that my thesis can act as that sort of catalyst too. I hope that the reader can honk and cheer and move on changed, and if not changed, then more aware of the bodies that aren't our own and what they're forced to live through. The images on the screen here are pulled straight from my thesis. The fawn was painted after my encounters this summer. The black lines represent habitat fragmentation, and the abstract red images can be paw prints, blood spots, flower petals. Incorporating watercolor alongside nonfiction and poetry creates a dynamic experience, which is important for a few reasons. No two readers will journey through my collection the same, just like no two beings move along the same path in the same way. The dynamic experience to also showcase how roadkill itself is a dynamic issue. We're not just talking about bodies alongside roads, but intangible impacts that reach three-dimensional into the air, on the surface of the earth, and below the surface as well. We're talking about roadside trash and sparrows' wings adapting to avoid windshields and habitat fragmentations with no animals across the road. My thesis exists because I love the environment. I love Marquette and all the beings that live here. Expressing this love and fighting for the beings that cannot fight for themselves has created a hybrid thesis that tells the story of bodies. Bodies of water and bodies of land, human bodies, but more importantly, more than human bodies. The sound of body makes is exactly that. All the sounds that a body can be. Thank you.